To the second SAF RA seminar on the effects of the COVID-19 pandemic on health and safety at work. Today we have a presentation by Dr. Tung Pham. It concerns antimicrobial or anti-static effects of nano-sized materials in protective clothing for workers in industry. Professor Pham is from the University of Innsbruck. He works at the Institute for Textile Chemistry and Textile Physics. So thanks, Professor Pham. I will switch over to you.
Okay, good. Thank you very much, Eric, for the opportunity to disseminate a little bit our works on the web seminar possibility. So as Eric introduced, my name is Tung Pham. I'm working here at the Research Institute for Textile Chemistry, Textile Physics. And the subject of my presentation today is already mentioned, antimicrobial in protective clothing. And I will switch off now my camera. Yes, okay. Ladies and gentlemen, in my presentation today, I would like to give you an overview on the nano-sized materials in antimicrobial protective clothing. And the outline of my presentation is just consisting of 3 points. So I will give you first the general aspects of antimicrobial in textile, followed by a specific example on nano-sized materials as antimicrobial agents in textile, and then hopefully we have time for some discussion on the future challenges and perspectives. The first question we have is why antibacterial textile? As we all know, due to the large surface area and the ability to retain moisture, textiles are known as being conducive to microorganism growth. For example, bacteria, also fungi, and these can be found everywhere, and they are able to multiply quickly and of course depending on moisture, nutrients and temperature. Some examples, some bacteria population might double every 20 to 30 minutes under ideal conditions, between 35 and 40 degrees centigrade, pH value 5 to 9. Also means that one single bacteria can increase to 1 million cells just in 7 hours. So in particular, they causes some problematic issues on textile. The first one for sure, we all know the development of unpleasant odor, and the formation of this malodor is in particular important as when microorganisms grow, they metabolize nutrients such as sweat and soiling present and produce odor-active molecules. Just an example, the metabolism of Staphylococcus aureus is believed to generate 3-methyl to hexanoic acids, which causes the typical body odor. The second aspect is the fabric deteriorations. As microbes often attack the additives applied to textile, we might expect discolorants and loss of textile functionality or even mechanical property like elasticity, strength, and so on and so forth. The third point, and I think it's related very much to the protective clothing, is the risk of contaminations to the protected persons. For example, here clothes in inguinal and perineal areas soiled with urines, for example, have been found to promote the growth of Brevibacterium, Amoklea canis, E. coli, and Protos mirabilis, enhancing diaper rash and associated infections. We also know that over 75% of food infection is attributed to the nematophilic fungi, Trifidum intertricalia, and these pathogens. So this is a great concept of microbial contamination. Just for example, mainly for textile, the use of hospital, but also for the use in medical devices for health and hygiene care. But also we know in sport clothing, water purifications, animal feed, and also in the food industry. Now moving directly to the antimicrobial treatment of textile, depending on the agent used and fiber types, including the fiber composition, structure, and surface, there are different chemical and physical approaches in order to impart antimicrobial property to textile. Some approaches are based on the use of specific agents which already, especially in the case of synthetic fibers, can be incorporated into the polymer matrix by fiber spinning in the fiber spinning process. But most of the cases in textile, the antimicrobial agents can be applied in the during the finishing stage, which is valid for, let's say, the major number of both synthetic and natural fibers. We do have some basic requirements when it comes to the efficiency. In textile, of course, we need to have the very high efficiency in antimicrobial effectiveness, but it's also need to suit the textile processing process. For example, dyeing, finishing. It has to be able to resist, let's say, processes like laundering, dry cleaning, and hot pressing. So that means to have certain durability. And of course, it's also need to have a certain level of safety. For example, low toxicity to the consumers, no allergic, no irritation or sensitization to the skin, but also need to have a certain environmental profile. Should not harm also the appearance and property of textile use. Of course, we also need to ensure safe handling and environmental friendliness during the textile treatment process. Now, when we were talking about the antimicrobial mechanism and types, we know that antimicrobial textile may act by two different ways, either by contact or by diffusions. In the contact case, the agent is placed on the fiber and thus not dispersed, so it just acts as when the microorganism touch the textile surface. And the other case, which is diffusion, the agent is on the fiber surface, and it will migrate from the textile to the external medium to attack the microorganism. As we all know, living microbes, a bacterium or a fungus, presence of outermost cells were composed of the polysaccharide, more or less maintaining the integrity of the cell component and protects the cell from the extracellular environment. So the survival of the cells is really depending on the outer cell wall, but also beneath the cell on the semi-permeable membranes. This is very important for the micro organism to survive and grow based really on the cell integrity. Therefore, we have more or less two types of antimicrobial effects. If their effects has happened due to the inhibition of the cell growth, we call it biostatic. But if the antimicrobial agents kill the microorganisms, we would call the effects biocidal. And as the antimicrobial agents act in many different ways. The main mechanisms of actions can be classified in several points. I just want to highlight some points here. We are talking about damage or inhibition of cell wall synthesis. We are talking about the inhibition of the cell membrane functions, which is also important for the regulate the intra and extracellular flows of substances. We are talking about the inhibition of protein synthesis. We are talking about the inhibition of nucleic acid synthesis, which is related to DNA and RNA, due to the binding of some antimicrobial agent to the components involved in the process of NDA productions. And of course, there are some inhibition for some other metabolic processes. If we would just now divide all the agents in two groups, we are talking about leaching and bound types of antimicrobial agents. The leaching types means that the agents is moving from the surface in which they apply on and enter the microorganism, poisoning it and disrupt the life or cause lethal mutation. The bound types means that the antimicrobial agents are bounded to the textile surface and make the surface antimicrobial active. It works by rupturing the cell membranes by the direct contacts. These types of modifications give the durability of the effects, but as you just compare to the leaching types, we all can imagine that the leaching type of actions may have a much higher efficiency. There are some typical antimicrobial agents in textile, so I just list them here for the sake of complications. Some examples. So typically we are talking about quaternary ammonium compounds, which is a quaternary agents which carry a positive charge at the nitrogen in the solutions. And the antimicrobial actions is triggered by the attractive interaction between the quaternary ammonium group and the negatively charged cell membrane of the microbes. Once in contact with the cells, they might damage the cell membranes, denaturalization of the proteins, and the inhibition of DNA, and so avoiding multiplication of the bacteria. The second widely used agent is triclosan, which is odorless synthetic chlorinated bisphenol, which is not ionized in solutions. And these biocide agents mainly acts as by blocking the lipids biosynthesis. And triclosan is mainly used in many applications, including soaps, lotions, creams, deodorants, and so on and so forth. In textile, triclosan is mainly used in polyester, in nylon, polypropylene, and acrylic fibers. It has some big concerns because it may have some certain antibiotic cross-resistant strains. Metals, metal oxide, and metallic salts, non-essential metals, can be extremely toxic to the most microbes at exceptionally low concentration. But to that, I will come a little bit later when we are talking about nano-sized antibacterial agents. Chitosan, it's a natural and hydrophilic copolymer. The way it acts is generally accepted is that in the first steps is based on the interaction between the primary amine groups, positive charge and a negative charges on the microbe surfaces, and then chitosan can penetrate into cell wall, combine with the DNA, and inhibit the synthesis of RNA, preventing protein synthesis. It also has some problem in crew handling, depending very much on the temperature and pH, which is also very critical for the stock, which is chitosan. I skipped the regenerable and halamines and the polyhexamethylene biconiates for the sake of time, and just move directly to the last example on the natural-based antimicrobial agents. These are compounds extracted from herbs and plants, and in the last few years has been extensively studied as alternative therapeutic strategy to combat microbial growth in textile. Compounds are, for example, lectins, polypeptides, flavonoids, quinonoids, tannins, and coumarins. But looking into the literature, we don't know yet clear how the let's say what is the working principles of those compounds. Just as a hypothesis, peptides just affect several internal cellular processes, for example, DNA synthesis. But yeah, there is no clear clarification on the mechanism. Now moving to the nano-sized antimicrobial agents. There are some very prominent examples. As you can see here, I have listed here in the table. For example, we are talking about silver, copper, zinc oxide, and iron oxide, and all of them are based on metals. That's why I would like to talk a little bit more on that one. The most important part of this nano-sized antimicrobial metals, nanoparticles, is, let's say, apart from the particle size, there is also the zeta potential of the metal particles, and the explanations look like that the smaller particle size would enable the particles to penetrate more easily into the bacterial cell bodies, interfere with cell membranes, and subsequently lead to the loss of cell viability. And when it comes to the potentials, this is concerning the electrostatic interaction between the particles with the negatively charged bacteria surface, which is also promote the penetration. Let me move now to a little bit more detailed description of this example. Of course, silver is one of the most prominent nano metal particles as antibacterial agents, and the practically silver has been used since ages, and the development of nanoparticles based on silver is just kind of a logical step. The main benefit of nano silver particles compared to standard silver, I think, is laying on for sure the small particle size, but also leading to very high active surface, promoting a very good release of silver ions. And we all know that the nanoparticles has kind of multiple mechanism of antibacterial activity. As the most accepted theory is metal silver reacts with the moisture on the skin surface or wound fluids. Then silver ions are released and damage the DNA and RNA and inhibits their replication. Moreover, on the aspect, the interaction of silver particles directly with the bacterial membranes also promoted as one of the mechanisms. So here we have two things. We have the membrane disruption, we have the interfere with DNA, but on the third aspect, we do have also the generation of reactive oxygen species as one of the key mechanisms for silver as antimicrobial substances. Just one example, when we are talking about silver nanoparticles having a diameter of roughly 20 nanometer, already with 75 milligram per milliliter concentration of silver particles, the growth of, for example, E. coli or Vibrio cholera can be almost completely inhibited. The second example I would like to move on is copper as antibacterial agent, and we know that in comparison to silver, few studies has reported the antimicrobial property of silver, and all the postulated mechanisms are based on the very similar principle to silver on the action modes, but the precise mechanism is still unclear, and it's believed that copper acts by combining with the thiol groups of key microenzymes. So the mechanism is more or less postulated to the protein inactivation via thiol interactions. And it is not so easy to separate the effects between the copper ions released from the nanoparticles and the particle itself. But we know that both play important role by the antimicrobial effects of copper. One example here is that, for example, copper nanoparticles with the average mean diameter of 100 nanometer reduce the growth of E. coli and Bacillus subtilis by 90% at a concentration of about 30 micrograms per milliliter, and it could kill completely, it could inhibit completely the growth by moving to 60 microgram per milliliter. And there's some interesting comparative study I have found. Some group has a comparative study between copper and silver, and in some cases they even reported that copper has even high activity compared to silver nanoparticles with the diameter of roughly 40 nanometer. The next example I would like to give you.
Can we just be just a bit quicker on the remaining slides, please, Prof?
Yes, okay.
I'm looking at the time.
Let's move to, okay, so I will skip zinc oxide, I will skip iron oxide. The interaction between clothes and microbes, I think I can also skip. Now, let me move to the silver nanoparticles incorporation in clothing. Just a few examples. There are two ways of putting silver nanoparticles into clothing, either by impregnating the substrate with colloidal silver particles or reduction of silver particles from silver salts into the fabrics. The key issue of the topical application of silver is still the durability, and the second one is the concern of silver due to the potential risk to humans and to the environment. Now let me move to the one example we have developed in the past. As you can see here, we have reduced silver from silver nitrates, and looking into the micrographs, you can see that we have reduced and formed silver particles within the range from 25 to 300 nanometer. We also performed some durability tests, and here we can see that after 5 washing cycles, more or less we have reduced more than 80%, 70% of silver. But as you can see in the bacteria test here, we see some, we have very nice prevention of the growth of the bacteria. The second example I would like to give you is the copper oxide reduction on the surface of cellulose fibers. Again, here we use we form copper 1 oxide using the ligand exchange reaction with gluconide. And again, here we also perform tests on the antibacterial effectiveness of copper. And if you look into the table down here, you can see also here after 5 wash cycles, we also lost more than 80%. But the good thing is that even with a very low concentration of copper after 5 times washing, we still have a very good bacterial reduction rate after 1, 3, and 6 hours. And looking into the SEM micrograph, you can see that we also form copper particles here in the range of between 30 up to 500 nanometer. We also have been working now on the new techniques in developing techniques to deposit copper on zebra seeds, but I will skip this slide for the sake of time, just to explain that we try to replace the use of silver by copper because of just for the economic reasons, that copper is a cheaper materials compared to silver, showing very similar effect on the antibacterial resistance. Now, the last slide I would like to present is the future challenges and perspective. The first thing I would like to mention here is those metal nanoparticles show some effects on the antibacterial effectiveness in textile. We always still have to consider regulation on nanomaterials, including the biocellular products regulation on nanomaterials. So that means risk assessment by development is a must. Also important to define the application environment, which protection are desired. Is it for clean rooms application? Is it in hospital? Is it for worker in the industries? The next question I would like to put is, when is nano really needed? Do we really need nano structures? The next question is, do we need to modify the bark or do we need to modify the surface? And as mentioned before, the main problem remains the durability. So we need to work on the phase bonding between the nanoparticles and the textile treatment. Again, here we know the leaching and the bonding types. So we are talking about the balance between durability and the antibacterial effectiveness. Here we need to work on the interface chemistry. Could we also imagine something we call rechargeable antibacterial reservoirs to load the antibacterial agent again and again? We need to work on the technological concept for economical generation of nanoparticles and how to incorporate into textile. And there's very nice review I found. There is a publication on the fate of nanoparticles in the environmental cycles. This is something related for sure to regulation, but on the other hand also to the, I would say, the awareness of the environmental friendliness of all technologies we try to develop in the future. And with that, I would like to close my presentation. Thank you very much.
Okay, thanks very much, Professor Pham. That was a very interesting topic, interesting presentation. Do you have a quick comment on the, I asked you about the impact on the novel coronavirus. I don't know if you've seen any research on this topic. Do you know whether, for example, the nano-silver particles are effective against these particles, for example, if they are included in masks?
Mm-hmm. Yeah, I can give some comment on that one. Yeah, I did some very fast search on the subjects as when Alexander asked me to give the presentation. And what I found is silver showed show some effect on I would say antivirus. Let's say there's only one study I found at least to show evidence on the effect of silver on HIV. not on the COVID-19, on the novel coronavirus. The main problem for those things is, as I mentioned before, all the metals need some kind of moisture environments in order to release ions, which is very very effective to the mechanism. And I could imagine if they are talking about masks having zero particles, I would say this is more, I would say, more effective to the bacteria in the surrounding when you breathe, rather than directly to the novel virus. So the direct correlation to the novel virus, unfortunately, I could not find it as solid scientific evidence so far.
Okay, thanks. Can you comment on whether that, so if it is shown to be effective, would that be a reasonable use, for example, of nanosilver, firstly from a economic perspective in terms of cost, and then in terms of the environmental impact you mentioned?
Silver nanoparticles, as we know, is already in use, and I think, but for the time being, is rather limited to the medical applications. I think first for the cost reasons, and the second one, I think also for the reason that there are some, let's put it that way, the risk of migration of silver nanoparticles through the skin is rather very low. So I could not find evidence for that one, but there are still risks of inhalation, in particular when we don't have very well-bounded particles into the substrates. So there are also some concerns on, let's say, in the contamination to the environment, not in the sense of toxicity, but the concerns relate to the killing effects of silver, to the to the wanted or let's say to the beneficial microorganism in the environments. And in my opinions, as I said, I put it here for the discussion. What do we need? And I think based on the requirements on the specific applications, then we need to evaluate, is silver really needed, or can we also move to copper? Do we need nanoparticle size, or do we need a sub-nano or only micro particle size dimension of the active substances?
Okay, thanks. Are there any questions or comments from other participants? So I see Julia is raising her hand, or your hand. Please go ahead.
Thanks. Can you hear me?
Yes.
Okay, thanks. Julia Catalan from the Finnish Institute of Occupational Health. Yes, one comment about nanosilver, because as far as I know, silver is at the moment perhaps the most efficient antibacterial compounds used in the hospitals.
Yes.
So it could be much better that the use is really restricted to human diseases and hospital use, because the problem if you use in another product is that you can really have the problem of induced resistance in the bacteria. So in addition, what you have mentioned, that the bacterial could be affected, the beneficial bacterial, there is also a risk that even the pathogen bacterial could become resistant in the future. And thanks a lot for your talk. It was really interesting.
Yeah, I fully agree. There is also concerns in how can I say, cross contamination to the resistance of some bacteria outside the hospital usage. That's correct. Yes.
Okay, thanks. I know there were a few years ago there was some use of nanosilver particles in, for example, socks and sports clothing, and this led to a lot of criticism from environmental defenders, at least, who thought that, as you indicated in your presentation, a lot of the nanoparticle is lost during washing, so they were very concerned about the environmental effects on wastewater.
Yeah. That I think maybe just my last comment. So that's why I think as the trends, we try to move away from silver. So that's why we try now to develop different techniques based on copper, and this is, I think, the more economically and in our opinions more practical approach to the wider industrial usage.
Is copper less toxic? Sorry, is copper less toxic to humans? Because silver is, at least in large quantities, is known to make people turn blue and to kill them.
Yeah. There is a nice study on that one, yes. But as long as copper concerns for the time being, of course, we don't have any, let's say, solid study on the effect on human beings. But what we do here is we compare the effects on more on the antibacterial effectiveness between silver and copper. And at least as I've shown in some study, we found that moving to copper is much more application close compared to silver. As I think the colleague from Finland mentioned already, we should really think on the restriction on the hospital usage.
Okay, thanks. Any further comments or questions? Oh, it seems your presentation was very clear. Don't be shy, people. Can you maybe just come back, Professor Pham?
Speaking.
Please go ahead, Alexander.
So thank you, Professor Tung Pham, for your nice presentation. So we are talking about possible cause within SAF RA. So my question is, what could be the main research questions coming out from your presentation? Perhaps you can a little bit comment on that.
Okay, yeah. I think, as I put it here, I think the challenge is in perspective more or less defining the future research. I would say from the research point of view, we all know that nanomaterials have huge potentials, but the thing I would like to highlight here is for sure that we need to consider the risks. So that means whatever we develop, we have to do risk assessments, and risk assessment for sure will be a part of future research. And also interesting for me to do some small research on the definitions of biocellular nanomaterials. They are not so much, and I think I would highlight the interface chemistry on the nano-sized applications was one of the key aspects for the future research, next to the risk assessment, but also on the environmental impacts of nanoparticles. This is something I think is not new, but for the time being, I haven't seen a very solid solution for that. So I would recommend to continue to that perspective when it comes to nanomaterials in the future.
Okay, and for sure, question is what can have effects of these nanoparticles on the COVID virus?
Yeah. For the time being, I think there's not much research on that one, as I mentioned before. The only study I found on the silver is on the virus HIV, and this is something I found in the scientific literature. But all the rest, I think it's more or less how can I say, people have tested it, and to the right, to the left, in the center, but a solid scientific base, I think, is missing.
And can you think about that? Also, the pharmaceutical industry is working on that topic, or could be interested.
I think the question. It's a little bit out of my comfort zone on my competence course. but I would say when the whole globes looking onto the I would say onto the how is it, on the prevention methods of the COVID-19, for example, what is it now? They call it even medical substances to heal the lung diseases, or to develop what is it now? Sorry, I get a blackout now on too many the name. I'm not so sure on nanoparticular effects on the viruses. I think this is for sure. We need biologists. We have microbiologists. We have pharmaceutical scientists. Yeah, to look into the structure of the virus for sure. This is something we need to understand, and then we need to look into the effects of what we have seen in the bacteria. If we can make some correlation and some parallel development to the virus structure, this is something we need to do.
Can you come back? You skipped over a slide, your slide 12 on interaction between clothing and microbes. I wasn't familiar with this, and I think it might be of interest to some participants. Can you perhaps comment that briefly, please?
Yeah. This is something I just tried to put. It is following. Yeah. As I mentioned before, we know normally the interaction between the clothes and microbes is a function. Yeah, more or less of the microbes in order to adhere to the fiber surface. Normally they form a kind of biofilms, and this depends for sure on the fiber hydrophilicity, the levels unbound water, surface water in the fibers, and very important also the susceptibility of the polymer to degrade. Yeah, that means to hydrolysis by the enzymes. And then I split it down a little bit into 3 parts. Just for example, when we are talking about natural fibers, which is more or less cotton, wool, and regenerated fibers like cellulosics, viscose, rayon, or nylon cell types, those fibers are hydrophilic, so that means they absorb a lot of water. They have rough surfaces, and by that they promote microbial adhesion. And also their structure is more susceptible to the hydrolysis, so that means they are easier, yeah, to be degraded. And then there are some classifications. People found that wool, for example, yeah, are more susceptible to bacteria, while cellulosics means cotton more to fungi. And then we have the other classes of fibers, which is synthetic. So we found it's like polyester, nylon, polypropylene, more on the functional clothing, as we said, yeah, in the outdoor clothes, sport clothes, running shoes, socks. They are more hydrophilic, and they have, due to the production technology, they have smoother surfaces, and by that they hinder microbial adhesion. And their structure are less susceptible to the enzymatic hydrolysis. The problem, what they all have is the following: they have the poor moisture absorption, but they allows the perspiration to pull, yeah, the moisture in the interstitial spaces, and the residues are susceptible to microbial colonizations. And the volatile byproducts of this, yeah, based on the microbial metabolism, are carboxylic acids, amines, and aldehydes, which give you the malodor effect. So that means if you have a running shoes, yeah, maybe produced from polypropylene or from polyester, you still have this ugly body smells when you are running. So it doesn't mean that a fiber which are more hydrophilic will automatically cause less malodor. Yeah, that's just
okay. Thanks. Yeah. Very, very interesting. I wasn't aware of this. Okay, we maybe have time for one last question. All right. Well, I think it's time to bring this presentation to a close. Thank you, Professor Pham, for this presentation. The slides and the video will be available on SAF RA's website, also on our YouTube channel, which we have opened recently. So thanks to everyone for the participation. I'm going to stop recording now.
Thank you very much.